Okay, so mitosis basically we want to produce two nuclei, not two cells. Be careful, guys. We don't produce two cells at this moment. We produce two nuclei containing chromosome identical to the parental nucleus, begins at the ends of the G2 phase. So between the G2 and mitosis, we do have a phase checking there called G2m checkpoint. G2m checkpoint is very important. G2m checkpoint basically to check everything okay or not, get ready, then only we continue with the mitosis. Are you clear? Okay, so DNA repair may happen at G2m. Okay, so G2/M. That's called a checkpoint there. So mitosis is a continuous process. This a bit of problem here is we don't have a very distinct, clear-cut point saying that, oh, we have done this particular phase. Are you clear? It's a continual process, the mitosis. So basically, even though we separate them into 4 phases, prophase, metaphase, anaphase, telophase, or most of the time I call it as a PMET. P stands for prophase, M stands for metaphase, A stands for anaphase, and T stands for telophase. So we call PMET. So even though we separate the process into 4 these phases, but we don't have a very distinctive, clear-cut, okay, we have done the prophase, then we continue with the metaphase, because they are overlapping. It's a continuous process overlapping. So it means that prophase and metaphase overlap, metaphase and anaphase overlap, and anaphase and telophase also overlap. So we have to actually find out the distinctive characteristic of a particular phase. Basically, when you see the particular characteristic, you know that, oh yeah, this is metaphase. Oh yeah, this is anaphase. So what are the distinctive characteristics? We actually focus onto the behavior of the chromosome, which we're going to discuss in detail later. Okay. So if you look at this figure 11.10, okay, summarizes the process of the mitosis. If you look at this diagram, okay, so we have the interphase. Interphase, basically, you won't see any chromosome yet. Can you see the chromosome? You won't be able to see the chromosome. What you see here is a chromatid. Can you pick up, okay, this is 1 chromosome, 1 chromosome, 2 chromosome, 3? Impossible, because at this moment, they are in the form of 10 nanometer fiber or 30 nanometer fiber chromatid. But once the cell progresses, finish G2, the cell progress to prophase. Then what happened here is the chromatin now start to condense. Are you clear? Chromatins condense onto this protein scaffold to form this, what we call the metaphase chromosome. So in this diagram, we have 4 chromosomes, right? 2 green, 2 red. So 1 green indicates the 1 set, 2 pink color or red color or pink color will be another set. So it's a somatic cells 2n equal to 4. In this diagram, you can see that it's 2n equal to 4. Okay, so what's the property of the chromosome here? This chromosome is termed as metaphase chromosome. Okay, metaphase chromosome distinctive characteristic, they have 2 sister chromatids joined at the centromere. Can you see that, guys? Each chromosome, they are butterfly, got 4 arms here. Okay, so 1 sister chromatid, another sister chromatid joined at the centromere. So prophase, this distinctive characteristic of prophase actually do divide into 3 substages, which you are not required to know. We have early prophase, prophase, and prometaphase. Prometaphase is overlapping between the prophase and metaphase. Are you clear? So because, as I said, that it's not a distinctive event, say that, okay, now done the prophase, so I proceed to the metaphase. It's impossible for us to pinpoint where is the stop of the prophase and beginning of the metaphase. Okay, so that's why sometimes scientists actually divide it into early prophase itself, and also metaphase. Are you clear? Any questions? So for us, what is a distinctive characteristic or the behavior of the chromosome? If you look at these structures, first thing, the chromatin, okay, condense to form the chromosome. Shorter, visible. Are you clear? In this case, because they condense very thick, right? The structure very thick, very visible structure, because the thickness or the diameter now increased to 700 nanometer, which we can actually view them by using the light microscope, but with the proper stain, because chromosome colorless. Okay, with the proper stain, we will be able to see the structure of chromosome. Are you clear? So when we can see chromosome, always remember when you use the term chromosome, we only can see chromosome during mitosis or meiosis. During this period of time, then your nucleus division takes place. Therefore, you have the chromosome. Okay. Now, next, what's a distinctive characteristic of prophase? Now you can see that during the prophase, you can see this. Okay, this is an animal cell. So animal cell will have this what we call the centrosome or centriole here. So centriole will migrate. Centriole will migrate to the opposite directions. Can you see that one going this way clockwise, another one going anti-clockwise, right? To the opposite pole, the movement, they start to move. When they start to move, they also organize the spindle microtubule. So they organize the centriole. Still remember the function of centriole to organize spindle microtubule. So what actually happened here is when they organize this spindle microtubule, this microtubule continue to form and attach to the centromere here. Are you clear? And this centromere have this kind of protein called kinetochore protein. Kinetochore protein allows the spindle microtubule to attach, okay, to attach to the kinetochore. And then what actually happened? You try to imagine, I am the chromosome forearm, okay? And my center here is the centromere, okay? So centromere here, you have the kinetochore, the protein, the binding site. So when centriole start to organize the spindle microtubule, continue to form. So this spindle microtubule going to attach to my kinetochore. Then it continue to grow, right? You try to imagine it's something that continue to grow and push me, continue to push me until the center of the cell, which you call as the metaphase plate. So therefore, you can see that the cell now, the chromosome can align at the center. This part we call as a metaphase plate. So when you see the alignment of the chromosome at the metaphase plate, then that is a signature of the metaphase. When you describe the events at the metaphase, you can see that centriole now already, or centrosome already reached the opposite pole. Spindle microtubule completely formed, attached to the centromere or kinetochore of the centromere, then align the chromosome at the metaphase plate. Okay, now metaphase plate is an imaginary plate. It's not a real plate. You won't see it. It's an imagination plate. So basically, it's something like this is the cell. At the center, normally at the center of the spindle, you have your chromosome aligned. Are you clear? So this part is known as the metaphase plate. Okay, characteristic event when you have all chromosome now aligned at the center or equator of the spindle. So therefore, it forms this kind of features. So after metaphase, you can see that cells start to elongate, become more and more longer. Can I see that? Longer. And what is the characteristic events here is this spindle microtubule becomes shorter now. Okay, this spindle microtubule becomes shorter. Now, what actually happened here is, look at this, our metaphase chromosome. Okay, look at this metaphase chromosome. And you have this spindle microtubule. You try to imagine, this green color spindle microtubule becomes shorter. When it becomes shorter, what will happen? It's something like you have this protein like this, attached to two strings. These two persons pull the string. So it's going to divide the centromere. Do you get it? It divides the centromere. So when it divides the centromere, what will happen here is centromere will move first. So you can see this something like this kind of structure. With the centromere move first. Can you see that? The spindle microtubule becomes shorter. Okay? Because of the shortening of this spindle microtubule, therefore it divides, not split, not break, it divides it. So divide equally, okay, the centromere. So then we divide this centromere. Now you can see that no longer called metaphase chromosome anymore. So when you don't see the metaphase chromosome, there's the end of the metaphase and the beginning of the anaphase. Are you clear? Okay. So the anaphase is a characteristic how you can see that sister chromatid now move towards where? Move toward the centrosome. You realize that? Sister chromatid. This structure is known as sister chromatid. No longer called as the chromosome. This is the metaphase chromosome. This is the sister chromatid. So sister chromatid, the pair of sister chromatid now move, okay, towards the opposite end. Okay, move towards where? Towards the centriole, away from the metaphase plate. Due to what? Due to the shortening of the spindle microtubule. Are you clear? Okay. So telophase, telophase, basically you can see that now this sister chromatid already reached at the opposite pole, and now they start to uncoil, form back chromatid. Nuclear envelopes start to reform. Can you see that? Okay, and you can see that the formations of something like this called cleavage furrow. Okay, the formations of this cleavage furrow. So this cleavage furrow, they later will join together in the cytokinesis, therefore forming two cells. Are you clear? Okay, so this is an overview. Later, I will go into more detail so that you will be able to see the entire process. Clear? Okay, so each phase, you have the signature events. Therefore, later we're going to discuss it in detail. Okay, so I stop the recording first. So first of all, let us start with the interphase. We start the interphase. So during the interphase, what actually happens? Okay, so now look at this. We have one cell. Okay, so this is during the G1. Okay, during the G1. This is not mitosis yet, not mitosis, during interphase G1. So this G1, you can see that you have the nucleus. And the DNA actually in the form of, okay, coiled to A with the, okay, DNA. One long, one short. Are you clear? Can you see that? And then we do have the centrosome. So I draw it as a centriole. Can you see that? Okay, so in this case, guys, now, if I ask you number of DNA per cell, so this one equal to 4. So 4 DNA. Are you clear? Now, if I ask you a number of chromosome per cell, now this is the confusion part, because we do know that during interphase we don't have chromosome. What we have is chromatid. So actually, the word chromosome here refer to chromatid. Are you clear? So the chromatids now also equal to 4. You get what I mean here? Okay. So what are the main characteristics of G1? So G1, basically the process actually is that they are very they are metabolically very active, particularly involved in the protein synthesis. So again, put a asterisk here. Oops, sorry. This is not mitosis yet, okay? This phase is not mitosis. Okay, so means that this actually is a 10 nanometer fiber or 30 nanometer fiber chromatid. So what actually happens now, the cells will go through the S phase. So where in the S phase, one of the very important event does take place here, DNA replicate. Okay, semi-conservatively. So means that what we are going to have now the cells, the nucleus. Now you can see that chromatin, okay, replicate. So red color chromatins now replicate. Blue chromatins actually replicates, okay, the DNA. Okay, so centriole still remains at this side. Okay, so this is during G2. Haven't started the mitosis yet. Okay, so if I ask you in this case, number of DNA molecule per cell, then now equal to 8. Double up because DNA replication takes place. From 1 molecule, get 2 molecules. So 4 molecules, therefore times 2, you have 8 molecules. Clear? Okay. But if I ask you number of chromosome per cell, answer still 4 because they join together, they are together. Are you clear? It's still 4. But again, as I say that, the word chromosome here is not really correct to describe. Actually, we refer to chromatin. Okay, so number of chromatin per cell still equal to 4. And the signature events that take place here, I use the black color. Okay, so again, continue with the protein synthesis take place. Again, the signature events here, protein synthesis. Clear? Okay. So prophase, okay, the signature events of the prophase, you can see that the cells, okay. Now, if I go back to this interface, you can see that actually in the nucleus here, we have the nucleolus. Nucleolus still remain intact here. Okay, you can see the nucleolus. Are you clear? Okay, but now during the prophase, nuclear envelope now disintegrated. We break this nuclear envelope or nuclear membrane. Okay, and due to the condensations of the chromatin to form the chromosome, now you can see that we form the metaphase chromosome now. Are you clear? And one of the very important events take place during this G2, we have also duplications of the centrosome. Are you clear? Duplication of the centrosome. So now this centrosome or centriole start to move to the opposite pole. Where is the nucleolus? Nucleus now disappear already. Can you see that? The term we use start to disappear. So it depends on which phase. It depends on which, prophase, early prophase, or what we call the pro-metaphase. Okay, so let us do the analysis here. So first, what is the number of DNA per cell? So per cells, we still have 8 DNA. Number of chromosome per cell is 4. So this chromosome actually refer to metaphase chromosome. Are you clear? Okay, so first we look at the behavior of the chromosome. So what is the behavior of the chromosome? The event take place. So first you can see that chromatin. I summarize here only, I won't write in detail. Later in the notes, I will show you the detailed answer. Okay, are you clear? Okay, so chromatins condense, okay, into chromosome, okay, which is thicker, shorter, and more visible form. This is the behavior of the chromosome. Okay, now how about nucleolus? So start to disappear. Okay, start to disappear. How about nuclear envelope? So nuclear envelope now start to be disintegrated. Now it's not disappeared, it's just that the entire nuclear envelope now becomes small vesicle. Are you clear? So we use the word start to disintegrated. How about centrosome in animal cells or centriole? So centriole now first they move to the opposite pole. So what they do, they organize spindle microtubule. So what happened to the spindle microtubule? In prophase, so start to form. Okay, a spindle microtubule elongates. Okay, can you see that? And last, plasma membrane. Nothing happened to the plasma membrane. Okay, so this is the characteristic event. Okay, it's too small. Now, guys, sorry, I think I can do. I try to move some of the notes to this side. Okay, make it bigger. Okay, clear? This is what actually happened during phase. Okay, a summary. Yeah, later we go. I mean, in the notes, I will go into detail. Okay? I think tomorrow we can do this. Okay. Any questions so far, guys? Okay, yeah. Have it done? Taking a photo of the prophase now. So this spindle microtubule, when they start to form, it becomes longer and longer. Kind of spindle microtubule. So when it become longer and longer, what actually happens? They're going to attach to the kinetochore of the centromere. Are you clear? So then what actually happen? You're going to push the chromosome towards the metaphase plate. Okay, so now we enter into the metaphase. Okay, we enter metaphase, the characteristics events that take place now, you can see that. So when metaphase, nuclear envelope completely disintegrated at this phase. Can you see that? Nuclei completely disappear. Okay, centrosome and centriole, they move to opposite pole already now. They reach the opposite pole. Okay, how about my chromosome? My chromosome now actually align at the metaphase plate. So you can see that I have the red color chromosome. Red color chromosome. It's not center, sorry. Let me make it more center. Okay, blue chromosome. I have to make it smaller. Okay, are you clear? Okay, so you can see that they align at this part. So this region is known as metaphase plate. It's an imaginary plate. Okay, and we do know that at the center of the chromosome, you have the centromere. And then spindle microtubule actually attached to this. Okay, kinetochore of the centromere. Can you see that? Okay, so now the question is, how many DNA? Number of DNA per cells. Sorry, per cell. So the answer is still 8 because we didn't do any divisions, correct? Okay, so how about number of chromosome per cell? So in this case, still 4 number of chromosome. So actually the chromosome here again refer to the metaphase chromosome. Okay, we refer to metaphase chromosome. So what is the behavior of the chromosome now? So chromosome, so now they align. Okay, they align themselves at the metaphase plate. Okay, behavior of chromosome, now they align at the metaphase plate. And one special event here is, so the kinetochore of centromere are attached to the spindle microtubule. Are you clear? This is behavior of chromosome. Now, how about this nucleolus? Nucleolus completely disappear. Nuclear envelope completely disintegrated. Okay, then centrosome or centriole. So what they do, they now reach, okay, the opposite pole. Okay, of the cells. Spindle microtubule. So in this case, completely formed. Okay, they are completely formed and they attach to the kinetochore of the centromere. Okay, so how about the plasma membrane? Nothing, no event associated to plasma membrane. So this is a signature of the metaphase. Okay, not guys? Simple, right? Okay, I just summarized the major events here. Okay, the small detail we're going to talk about it tomorrow, right, during our online class. Okay. Any questions so far? No, okay, now next. When we are done the metaphase, so the next part we're going to have, that after the metaphase, we have the anaphase. So anaphase, you can see that the cells start to become elongated, but no cleavage furrow yet, yeah? Oops, sorry, wrong. Because of my direction, so it should be longer this way. Okay, so my centriole or centrosome still at the opposite pole. Okay, so you can see that my chromosome now move towards. Okay, but we don't call this as chromosome now. Okay, so centromere now move first. Okay, attached to the spindle microtubule. So you can see something like a finger structure, something like finger like this, they move. Okay. So where's the metaphase plate? So here is the metaphase plate. Okay, it's imaginary plate. So again now, first, number of DNA per cell. How many? 8. Okay, number of DNA per cell still 8. Number of chromosome per cell now equal to 8. Double up. Because the chromosome here actually refer to sister chromatid. Okay, now this part I'm going to talk about spindle microtubule first, the behavior of spindle microtubule. So what actually happened to the spindle microtubule? So shorter. It becomes shorter. So when the spindle microtubule becomes shorter, what's the behavior of the chromosome? So in this case, I say that due to the shortenings of the spindle microtubule, now centromere. Okay, the shortening of the spindle microtubule divides. Okay, the centromere equally. So because of divide the centromere equally, then our sister chromatid now move towards, okay, respective centrosome. Okay, respective centrosome or centriole away from the metaphase plate. And very important to indicate here, centromere move first. Okay, very important to state this, centromere move first. Because why? Because they attach to the spindle microtubule. So when a spindle microtubule becomes shorter, you actually pull. You try to imagine it's something like this. Okay, can I have 4 pencil? Okay, pencil, pen doesn't matter. Now look at this, how I'm going to do. This is a chromosome. Are you clear? Chromosome, right? Okay, one sister chromatid, another one sister chromatid. So it will be something like this. So centromere attach the 2 sister chromatid together. Okay, so metaphase plate, they align at, so during metaphase, they align at the metaphase plate. What happened if the shortening of the spindle microtubule? So divide the centromere equally. Can I see that? But when they move, because it's cytoplasm, it's not the air. Cytoplasm had a resistance. So it means that when I move towards the centrosome, because of the resistance, so you can see that centromere actually move first. Do you get it? So this is a characteristic of the anaphase. Do you get the idea? Okay. Thank you. So, okay, so that's why the concept of centromere move first must be there. Okay. So now next. After the chromosome, how about the nucleolus? So remain. Okay. Disappearance. Okay. Remain. Still completely disappear in this case. Nuclear envelope still completely disintegrated. Means that nothing happened lah. Basically, nucleolus, okay, already disappeared. Are you clear? Nuclear envelope already completely disintegrated. So it's there. Centrosome or centriole So they are still remain at the opposite pole. Okay, how about plasma membrane? Plasma membrane still have nothing events, okay? But the cells now become elongated. Are you clear? The cells become elongated. So a bit small, right? Sorry, okay? So we may take the photo if you need. Maybe I can move this nuclear envelope. Not here. Okay. Okay, guys. So I stop at this anaphase first. So tomorrow we're going to continue